How's it going, everyone? It's Chow here, and today we are going to talk a little bit about mutations, so let's get started. Okay, mutations. So mutations are very fascinating because they're actually nothing more than changes in the DNA sequence. I know it seems pretty complex, but it really isn't. It's just some kind of change in a DNA sequence. So there are kind of two types of mutations that are found in the two main groups of cells that we have in our body. The first is the somatic cells, and in somatic cells, those are our normal body cells. And mutations in somatic cells often are due to problems in mitosis. And these results can often cause issues such as cancer. Whereas on the other hand, we can also have mutations in our germline cells, so in our sex cells. And if we have mutations in our sex cells, these can often be passed on to our offspring, which can be very, very problematic. Or it might be good depending on the situation, which we will talk about later on in this lecture. But in terms of mutations, there are kind of two main groups of mutations available, and the first one is the point mutation. So the point mutation affects only one specific point in the sequence of, for instance, the DNA, or ultimately, eventually, the mRNA sequence that's resulting from the DNA. So what can happen is that in a point mutation, there can be a type of mutation called a silent mutation. So a silent mutation is one that basically has no effect on the transcribed protein. So remember, think back to our genetic table, right? Our amino acid table. And you can see that in a lot of amino acids, there are a bit of redundancy within the code. So due to the redundancy of the genetic code, one single point mutation might not have an effect on the amino acid that's going to be put into that place, which makes it going to be a silent mutation. We can also have a missense mutation, which is basically a single point mutation that causes the change in the amino acid. So one single point mutation causes the amino acid, for instance, maybe a glutamate, to be switched to a valine or something like that. So that's going to be a missense mutation. We can also have a nonsense mutation, which is basically a mutation that causes a premature stop codon. So sometimes what happens is you have one mutation that causes a stop codon to be in place instead of a normal amino acid, and therefore you cut the entire polypeptide much shorter than potentially what it normally should be. So that's a nonsense mutation. So a nonsense mutation, again, is a stop codon. A missense mutation is basically a change to one single amino acid. We can also have a frame shift type of mutation, and a frame shift mutation is basically insertions or deletions of one or two nucleotides, but anything that's not a multiple of three, basically, and it's basically going to throw off the entire reading frame. So effectively, you still might make a relatively long polypeptide sequence, but because the reading frame has been shifted and changed, you're ultimately going to be putting in potentially the incorrect amino acids because of that shift in the reading frame. Remember, the reading frame is, each individual three codons all the way up to a stop codon. So if you add in something and maybe you shift the reading frame by one or two letters, that can have a very strong and negative impact on the resulting polypeptide. So these are the point mutations. There are also other types of mutations that are much larger. So we can have chromosomal mutations, which can basically alter segments or entirety of chromosomes. In terms of chromosomal mutations, there are kind of four main types. We have a deletion. So a deletion is basically a segment of the chromosome being removed. So that's pretty straightforward. We can have inversions, and inversions are basically sections of the chromosomes that are going to be flipped. So this top part is going to ultimately come down to this bottom part, and it's basically going to be on the same chromosome, but you're just basically almost removing a section of that chromosome, flipping it, and then putting it back in. So that's an inversion. Duplications can also arise in chromosomal mutations. And what a duplication is you have a section of the chromosome that's going to be duplicated and put back. So we have a section of chromosome here. You duplicate this section and then you put it into that same chromosome, which pretty much lengthens the chromosome and adds in the same sequence. So it arises potentially in recombination events where chromosomes don't necessarily break in the same place. We can also have translocations, which is the final type of chromosomal mutation that's relatively common. And a translocation is basically genetic swapping between non-homologous chromosomes. So you have a section of the chromosome on this particular pinkish chromosome that's going to be swapped with this particular section on this yellow chromosome. And this can basically create novel pathways and novel chromosomes, which can be very detrimental, or it can also be somewhat beneficial in rare instances. So think about the different types of chromosomal mutations as well as the different types of point mutations. So that's something definitely good to keep in mind. So what exactly causes mutations? Well, sometimes mutations can be due to more spontaneous types of damage. So tautomerization, and then of course, addition or loss of certain functional groups. So you lose an amino group through deamination, or you add in a methyl group to a particular base, and these can ultimately cause problems with base pairing. So you have originally in this enol form an OH group that's pointing up, and then in this particular keto form you have a ketone in that area, and that can basically affect how bases are going to be paired. You can also just simply have error due to DNA polymerase kind of messing up during replication, so it inserts the wrong nucleotide, and then your cell wasn't able to recognize that incorrect nucleotide and fix it, and so it just stays incorrectly paired. You can also have imperfect meiosis, which affects sort of larger chunks of genetic material, because oftentimes you might even have an extra chromosome. So through issues such as non-disjunction, you can gain an extra chromosome in the gamete. So in this instance over here with this red chromosome, normally the sister chromatids are going to be pulled together by the spindle. But if non-disjunction occurs and that sister chromatid is, those sister chromatids aren't going to be pulled together, you effectively have one particular cell that's going to lack an extra chromosome and one that's going to have a gain of an extra chromosome. So one cell has one less chromosome than usual and one has one more than usual. And through non-disjunction, you can cause certain genetic illnesses and certain problems, including, for instance, trisomy 21, which can cause Down syndrome. So these are more of spontaneous damage. You can also have more physical induced mutations that are caused by chemicals or mutagens. So chemicals can alter bases, and that can cause mutations. So in this instance, we have UV light that's striking two thiamines that are next to one another, and they can actually cause the thiamines to dimerize, and instead of pairing with the bases on the opposite DNA strand, the dimers basically pair with each other. That can have a very negative effect on replication and stability. In addition, you have chemicals that can add in groups to bases, and you can also have a situation where you have radiation maybe that just kind of slices apart different DNA strands, and that ultimately can also cause issues. So sometimes you can have mutagens also that kind of intercalate between the DNA strands and break down that base stacking interaction, and that can ultimately change the stability of the DNA and ultimately cause problems with ultimately potentially mutations. So there you go, causes of mutations. Now, what about the effects of mutations? So mutations don't necessarily actually cause bad things all the time. Sometimes mutations can provide genetic diversity. In fact, mutations are basically the basis of all genetic diversity on lineages of organisms. So if there's a mutation that has some kind of a selective advantage, over time you might see that particular mutation being passed on. or the mutations can do nothing. So you have silent mutations, remember those? Those silent mutations don't really have any sort of selective advantage on the organism, but they also don't have any selective disadvantages. So over time, those mutations might build up in the organism simply because it doesn't have any positive or negative effects. And finally, last but not least, everyone's favorite, mutations often cause genetic disease and issues. So mutations can be sort of good potentially in the long term, it can be neutral, or it can be very bad. So in terms of genetic disease, mutations can cause various problems, including things such as inability to transfer oxygen from one place to another. So obviously we're talking about sickle cell disease, which can eventually cause sickle cell anemia. And this is only through a point mutation. But obviously with large deletions or whole chromosomal abnormalities, you can cause other issues, for instance, again, such as Down syndrome. So with this particular example, we have sickle cell disease, which is basically one single point mutation that instead of causing a glutamic acid or glutamate in that particular sequence of amino acids, you ultimately cause it to be changed to a mutant valine molecule. So glutamate versus valine. Glutamate is going to be sort of an acidic amino acid. It has a relatively low pKa, whereas valine is more of a neutral hydrophobic amino acid. So these can often cause issues in regards to how the proteins are folded, and obviously we can see such in the resulting shapes of the red blood cells, and sickle cell disease can obviously decrease oxygen flow, and that can cause many great problems. Okay, now in terms of genetic diversity, however, we have to keep in mind that all individuals have a unique DNA sequence. So we can use fingerprinting methods to basically analyze a pattern of mutations in individuals to determine the relatedness between those individuals. So maybe like a paternity test, if you want to think about it that way, or maybe to figure out whether someone did a crime or not. So we often will look at things such as polymorphism. So a SNP, or a single nucleotide polymorphism, is basically a point mutation variation in certain spots in the genome. We can also have STRs, which are short tandem repeats, and STRs are basically repetitive sequences next to each other, oftentimes in a non-coding region of a chromosome. So genetic diversity is definitely present in individuals, and we can definitely use modern technology to kind of figure out some of that genetic diversity through various methodologies. Of course, when it comes to genetic disease, obviously one of the things that we want to do is to try to mediate it or potentially even cure it. And there are basically two methods that we like to use today to approach genetic disease screening. The first method is through phenotypic screening, where you're basically screening for activity or protein problems. So you're screening for that particular protein or product in the process, the phenotype. The other way is to actually use genotypic screening, which is basically going in and testing the DNA itself for the presence or absence of certain known mutations. Okay, treating genetic disease obviously is probably one of the most prominent fields today in biomedicine. And it's not necessarily the most straightforward one, because genetic diseases, as of now, can't really be 100% cured. But we can treat it to try to make the person who's suffering from it live a more successful life. And one of the ways we can do that is by actually modifying the phenotype. So this is modifying the phenotype of the issue. And there are some ways we can do it, including restricting the substrate. So in PKU, which is an illness where basically individuals have issues metabolizing, especially in young individuals that have issues metabolizing phenylalanine, which is an amino acid in their diet, you can basically decrease phenylalanine or basically remove phenylalanine completely from their diet to treat that illness. We can also use metabolic inhibitors. So if there's a mutant protein that's really, really active, you can use a metabolic inhibitor to basically compete with the substrate of that protein, and you can potentially inhibit that protein. You can also use basically supplemental information, so basically supplying something that's missing from the body of a person who's suffering from a genetic disease. So very common is if the body doesn't have enough insulin in diabetics, you can basically inject insulin into the body to ensure that those individuals can store glucose, for instance. And obviously this particular method is by simply supplying the missing protein. So this is a more phenotypic approach to treating genetic disease. You can also have a more genotypic approach to treating genetic disease, and here is probably one of the most prominent fields within treating genetic diseases is using gene therapy. So you're actively going in and replacing the defective gene. So there are two main ways of doing this. The first way is with an ex vivo method, which is where you take the cells out of the body, you put new genes into the cells in the lab, and then you put those cells back into the body. So ex vivo method. The other method is an in vivo method, which is basically where you use a vector, right? So you take a vector, you get the genes in that particular vector, and then you use that vector to inject the genes into the body, and hopefully you solve the issue. So in regards to approaching ex vivo and in vivo methods, there are also two main targets. We can target the germline cells, which is where you're inserting those replaced genes into a gamete, which can potentially affect all cells in the body of the offspring. The other method is with a somatic approach, where you're inserting only into the diseased cells, and that is going to have a more localized effect. But overall, through ex vivo and in vivo methods, whether it's in germline or somatic cells, gene therapy is a wishful method of medicine that can potentially replace defective genes, so you're going all the way back to the genotype and then replacing potentially the defective gene. That's very different from simply treating the symptoms. So instead of just modifying the resulting phenotype, you're trying to solve the underlying issue completely. And this is definitely a field of medicine that has a lot of research potential. But that's basically all I have for mutations. I hope you all enjoyed this lengthy discussion on issues with our DNA. And as always, hope you found it useful. Best of luck studying, and I will see you in the next video.